Thank you.
Seat, I'll just introduce you and then we'll wait for a few minutes because we have a bus coming. That bus just came and people are having breakfast.
Okay.
It will take a few minutes, so maybe we'll start.
No problem.
10:05.
Okay, I can even switch.
Good morning, everyone. I hope all of you are almost through with assignments or the projects that you're doing. It's coming assignments. Okay, good. So today morning, we'll have 2 talks. And the first speaker is Daniel Jost. So it's a pleasure to welcome Daniel. Daniel is a biophysicist at ENS Lyon. He has been working, doing theoretical studies of various biological systems. And he worked on different aspects of both chromatin as well as gene regulation. And for last several years, he has been working on modeling chromatin. So he will tell us today about the physics of phase separation in nucleus. Daniel.
Thank you, Ranjith. So, hello everyone. Ranjith asked me to make a large introduction about phase separation in general, because the background here is quite diverse. So I tried to make it. But of course, there might be some holes in the presentation, and I may assume that you know things that you don't know. Okay, so please interrupt me. But I think you are a good audience, and please ask questions during the talk. It's not a problem. So since a few years now, there is a lot of interest in biology about condensate or biomolecular condensate. So what is a condensate? I mean, it's not a really precisely defined notion, but you may see that as the presence of specific region in space where you have an enrichment of some kind of proteins or factor that may form specific membraneless kind of organelles. Okay, so sometimes people will talk about foci, okay, condensate, which is this kind of unknown large-scale definition. Sometimes people would say droplet, but droplet already have a meaning about a liquid somehow. Okay, so but generally it's better to talk about biomolecular condensate. And there is a various of biomolecular condensate inside cells. And along the years, the recent years, a lot of discoveries of these new organelles. And I mean, one of the first quantitatively studied was pig granules. In the nucleus, you have the nucleolus, okay, which is a part of the nucleus where ribosomal RNA is transcribed, and many kind of condensates, okay. And there have been also a variety of experimental characterization of this condensate. So when you do observe this kind of enrichment of a protein somewhere, the first thing is, of course, to observe it and to characterize the properties of that condensate. And I will not comment too much on that. Maybe Yuval will describe some of these techniques, I don't know. But there is a variety of techniques going from microscopy to sequencing to in vitro analysis to try to understand the physical but also the chemical properties of this condensate and their biological consequences for the cell. And regarding the functions, the biological function of this type of condensates, also a variety of hypotheses have been made about why it is important for the cell to have this kind of phases that are appearing. Not all of these kind of consequences have been proved experimentally, okay? It's more sometimes like concepts or functions that may exist in just in vitro, nicely characterized chemical systems, but in biological systems, sometimes it's quite hard to look at these functions. But for example, one of maybe of the most intuitive functions of this type of condensate is that locally you will increase the concentration of some proteins or some factors, some enzymes. Okay? And by locally concentrating the concentration, then you will become more efficient to perform the action this protein. So that's one possible function of the formation of this condensate, but as I see, there is a variety of possibilities. And as we are here to discuss about chromatin and chromosome, of course, inside the nucleus, there is also a variety of different condensates that have been characterized along the years. Here it's an example of some of them. Again, it's not exhaustive, and there are new condensates that are, I think, discovered every year these times. And just to focus on few of them, so there is typical condensates that are associated with activity, gene activity. And one well-known example is mediator, for example. We were discussing that yesterday. That may form, so mediator is an important protein for gene activation. And some genes are highly activated by enhancer. I think since yesterday you know what is an enhancer and how it might work to activate gene expression. And so the formation of this condensate around these enhancers could be also an important aspect of gene regulation by, again, increasing potentially the concentrations of transcription factors around genes. And there are also a variety of condensates associated with gene repression. Okay, so, and these are two famous examples. One is polycomb bodies that are associated with the repression of genes that usually change their status during development. So genes that are expressed at a given time window during development and that will become repressed after or before. They are usually regulated by these machineries that repress genes when they are repressed, and that also have this capacity to form this kind of condensate. And another well-known example is heterochromatin. And by here, heterochromatin, because again, people will talk about heterochromatin in various ways. This is what is called constitutive heterochromatin. So that's the chromatin that is usually around centromeres or around telomeres that really here repress constitutively genes that are behind. And this is associated with some proteins that I may talk about later. Okay, so again, so it's just a zoology of different condensates, also inside the nucleus. And of course, the most well-characterized, I think, organelles in terms of condensate in the nucleus is the nucleolus. So, of course, when you talk about condensate, as a physicist, it rings some bells, and this already points to some phase separation properties. So phase separation means that, that's a typical daily life experiment that you can do in your kitchen, right? If you mix oil and vinegar or oil and water, at a given concentration of oil, which is quite low for this type of molecules, very rapidly you will form droplets, okay, inside the water. And this is called phase separation because the oil wants to stay more with oil than with water and will form these droplets of oil. And this phenomenon is called specifically liquid-liquid phase separation in the case of oil and water because this is too liquid. And the droplets has the properties of liquid, and the kind of the rest, the solvent, the water has also a property of liquid. But phase separation is a general phenomenon. It doesn't mean between two liquids. It could be just a separation between two phases. And so, of course, people wanted to understand if the condensates that were observed in the nucleus, but also in the cells, were driven by the same properties or the same physics that drive the phase separation in your kitchen. And so, to address that, people use very simple theory first, okay, to try to understand this. And there have been a variety of, the study of phase separation in physics or in chemistry is since decades now, and more than that. And so there is a lot of theories that try to address this type of questions. And in the next slides, I will try to briefly introduce what is the most simple theory that may give rise to phase separation. So it's called the Fleury-Huggins theory. and it's very simple. So I will just try to explain. So now you start to be familiar with modeling for let's say the biologist in the room. For the physicists, maybe it's a little, you may have already seen that, maybe not already, but that's a typical simple model for explaining phase separation. Yeah, so to my knowledge, that's the most simple explanation. So the idea is very simple in that you are mixing two molecules, okay, two types of molecules. So oil and vinegar, for example, okay. And you want to understand if these molecules can form condensate. Okay. So here it's a specific example where the molecules are polymers, okay? But the model is quite generic, and of course it's even more simpler if you only assume that the molecules are just simple molecules and not polymers. Anyway, so you have two types of molecules. And you may assume that these molecules have some kind of interactions or energy of interactions between them. So reds and reds when they are close together. So this is an example where you have these particles on a lattice. That's the most easiest way, again, to define a space. Okay? As the theoreticians, you may discretize space into a lattice. And why not a square lattice? And why not this lattice in 2D? Okay? Again, this type of theory nicely apply in any dimension, but again, it's a nice way to illustrate that aspect. Okay, so you have a lattice like that, and you have these entities on the lattice. So either you have a solvent, which, for example, are the blue, or your molecules. Okay, so solvent could be your water in your daily experiment, and the red could be the oil, for example. And they are moving, they can freely diffuse on a lattice. Okay? And what drives potentially the agglomeration of these molecules into condensate is typically the interactions that these molecules have with each other, plus entropy, of course. But I think, again, since few days now, you have a notion of entropy. But you have at least these interactions between the molecules. Okay, so yes.
So here I see that you have two arrows. One is induced oligomerization, another is spontaneous. So this induced oligomerization would be something energy driven, or we are talking about non-energy driven?
Yeah, I think you can forget about the induced oligomerization. That's a typical. It's a one type of experimental pathway you can do to get a phase separation of polymers. Is that you mix monomers and then you induce the oligomerization, and then you may. But for the theory itself, it's not necessarily this type of induced oligomerization. You need the molecules. Here is just a way to form the polymers from the monomers, but it's not something that it has to be explicit in the model somehow. Okay. Actually, I will mostly talk about this. This is just let's say the last step of the theory where you want to talk about polymers rather than just monomers. But so far, I will introduce all this on the monomeric side, because the polymer properties is just a small step in the theory that is not adding qualitatively big things in the theory itself. Okay.
Just in this context.
Yes.
So people say, at least in the context of biomolecular condensates, you need valency, which is like multivalency and all that. So for a monomer, having multivalency is difficult compared to a polymer CC. So the polymerization, is it a necessary step, or is it? I mean, I understand that monomers can in general phase separate, but when it forms a biomolecular condensate, monomeric thing would phase the polymeric things would phase separate better, right?
So the polymer, well, you have two sides, because maybe you have an increased valency of interactions, but you have also internal entropy for the molecules, because now it can have internal structures. And you have also steric hindrance of the molecules that are on, and the packing somehow might be different if you are packing polymers, complex molecules, versus monomers. So in the theory itself, it's quite simple. You only assume that you have a given number of interactions with your neighbors, and that's it. You don't care too much, even for a polymer, you don't care too much about the exact numbers. And of course, this will have an impact on the quantitative details on how the phase separation behave. Okay, I will talk about this later, this phase diagram. But in the qualitative behavior is not really dependent too much on the exact details on that. I don't know if I'm answering the question.
Go ahead, yeah.
Any other questions about these steps now? No? Yes?
Can you raise your voice?
Is the phase diagram for a polymer system because it's asymmetric? For a monomer, I think it should be symmetric.
It should be symmetric, yeah, you're right. So to be honest, I picked this phase diagram somewhere on Google Image. But you're right that for a monomeric SiC, it should be symmetric in concentration, okay, because the system is purely symmetric. So it's likely, you're right, that the phase diagram here was done for a polymer. But I didn't make it, yeah. So I cannot tell you the details. It's just a qualitative illustration of this, yes.
One more question, like, is the transition you mentioned faster, or that's when you increase concentration? If you go from like homogeneous to the demix region by decreasing chi, is it second order or first order?
I will talk, wait a minute and I will describe this a little bit more, okay?
Thanks.
So any question about just basically the description of the model? It's just, okay, it's just a scheme, okay, an abstraction of what could be, I mean, that's the way we like to do in physics, okay? We start from very basic assumption. We try to make predictions, see if this is fitting with the experiment, and likely not, okay? But then we have to refine the model step by step, okay? But by doing that, we understand at least the grounds, and then we can decorate the models, and we understand every step. And this is maybe the first step for phase separation, the basic one. Then you can add, this is the first one, then you can add if this is a polymer or not a polymer, the complexity and all these type of things. But for this, okay, you only need very simple ingredients. One is that you assume that what drives this phase separation is the interactions between molecules, okay? So two red molecules would have a given attraction between them, okay, or repulsion, whatever, some kind of forces between them. So either they like to be close to each other or they don't like. Same for the blue and same for red and blue, if they are close to each other. Of course, naively, you have to say that if you have a phase separation, for example, between oil and vinegar, is that oil prefer to stay with oil rather than water. So basically, the strength of interaction between oil and oil molecule is stronger than the strength of interaction between oil and water. And the beauty of this theory is that all the behavior is mostly driven by one parameter. So all these details about interactions between molecules at the molecular scale, about the valencies of interactions between molecules, everything is hidden into just one parameter. This one, which is called key usually. That is just this one that drives the transition, plus, of course, the concentration of the different elements in the system. Of course, then it's easy to assume that if you have no red molecules, the system will behave very differently than if you have all red molecules, okay, of course. And so there is only these two parameters that drive the phase separation in this very simple scheme, okay? And I will not describe too much the details of that, but one key element here is that the energies are here in this kind of formula. So again, I will not describe too much, but just to tell you that all these microscopic details came here into these parameters, okay? And what is happening is the relative strength of interactions between red and between red and blue, more or less, okay? So this is relative, actually, okay? If I prefer to stay with red than with blue, I'm likely to have a phase separation with some condition. But the same, if I really don't like to stay with blue, I don't care to be with red, but it's not an attraction. It's like, I don't care to be. Again, I will have a phase separation between red because I really don't like to be with the blue. Okay, so it's not a matter of attraction, repulsion. It's just relative behavior. And another critical parameter is the temperature, okay, also. So as you increase the temperature, and that's also you know, as you increase the temperature, you tend to have less phases, okay? So the degree, if you increase temperatures, these parameters will decrease, and it's basically will perturb the phase separation, okay? Again, and so now what drives the phase separation is these parameters, okay? And what, of course, may drive this phase separation is a battle, but that's always the same usually in statistical physics. It's a battle between entropy and energy, okay? Energy is this interaction that wants to make this condensate, and entropy is the statistics, the randomness that wants to spread the molecules, okay, and not to form this phase separation. Yes? In this theory, the temperature is entering in the system like that. So I'm not sure to fully understand the question.
What I mean is like, so with chi, how chi will change the temperature. So depends on, I think, the mixing of the two is A and B component. Whether they compare LCST or UCST.
LCST.
Lower critical.
Okay, yeah. In this type of theory, you don't have this type of detail, of course. That's the basic theory you can have. Then you can build on that to describe this type of behaviors that you are mentioning, okay? Where you have freezing point. I mean, you have a variety of transition, okay, in a system like that. You have glassy transition also. I mean, there is a variety of phase transitions that may happen. This is just the simplest way of describing and the effect of temperature for the formation of condensate, okay? But again, the aim is not to be able to capture all the different transitions for a specific system. Again, if I'm mixing oil and water and I'm playing with the temperature, at some point my water will freeze while the oil is not exactly, or the reverse. Or, okay, then it becomes complicated, okay? But again, if I'm just interested in how I can describe a phase separation, that's the most simple things. And actually, it works not so bad for many simple mixture of different molecules, okay? But the very details, of course, of these interactions are important, okay? It's not that, and the temperature is very important. But again, qualitatively, everything depends on only chi and concentration, more or less.
That was the coordination number.
Yes, 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 exactly. So on this 2D lattice square, it would be 4. But yeah, exactly. So if you interact with more, potentially with more monomers around you, chi will increase more easy. So again, you increase the potential interactions, and so you increase the strength of phase separation, if I can say that. All right, so again, I don't have the time to fully describe the mathematical details of the theory, but with this very simple, and it's not so complicated, but it will take an hour maybe for you to understand fully all the details. But the nice thing is that with these two parameters, so this kind of chi and concentration, you can draw what is called a phase diagram, which basically it gives you for any value of these two parameters, if you can have a phase which is formed, a condensate if you want, or not. What does it tell you like that is that, okay, if chi is not high enough, you will never see a condensate forming. You will never see phase separation. So if, for example, the temperature is too high, as you may know, okay, when you increase temperature, you increase the entropic part of your system. And so particles would like to spread and to move and to occupy the space, okay? And so if temperature is too high, anyways, they will not like to be with each other. Okay? They will prefer to move. So you will never see a phase separation here.
So then in a biological system in a cell, temperature hardly varies.
Yes.
Right? So what would be the analog of temperature in biological system?
So as you see in this parameter chi, there is temperature. There is also the interaction. So you may play with interaction by post-translational regulation, for example, that will have an impact on the strength of interaction between molecules. So that's a way to have an impact on these parameters in the cells. Again, this theory is also very simple, but in the cells, there is other things that may act on the phase separation itself. So temperature is not important. It could be an effective temperature again here. I mean, I will not discuss this too much, but you may, even if you have activity in the system, so basically you have ATP-dependent activity, you may see that very simply as an effective temperature that basically increase the temperature of the system. So the degree of activity of a given process may potentially also have an impact like an effective temperature, because activity, let's say, naively, if you have activity, it's like increasing the fluctuation in the system, and so increasing the temperature. If you have low activity, maybe you will decrease the temperature of the system. So again, that's potentially a way in the framework of this very simple theory to have this change of these parameters in the cell. But I would say most intuitively that would be to play with the interactions between molecules, because we know that post-translational modification, for example, have a clear impact on this type of property. I think that's a whole good question. I don't think that really people have addressed that correctly, but that's because, of course, activity, ATP-dependent mechanism could be seen at an effective temperature, but it's not an exact increase of temperature because the shape of the fluctuations are very different than thermal fluctuations. So it's a very good point. But if you consider chromatin, for example, and you look at the motion of chromatin inside the nucleus, it has a diffusion, okay? You can measure a diffusion, okay, of that. I'm not an expert, so I will not detail, and I don't know, but you cannot, the coefficient of diffusion that you measure in the experiment, okay, cannot be simply related to temperature, like the actual temperature of the system, okay? Like, because in simple thermodynamics and out of equilibrium physics, you have simple relation between the diffusion of a molecule and temperature. It's proportional, okay? In vivo, you do not have this simple relation. So if you increase the temperature in vivo, you will not either have an effect on the diffusion or so that's why we mostly talk about effective temperature in vivo, but the exact relation, it's a very complex thing. And, I will not be the person to discuss that, but I'm sure people have tried to address that question.
So anything that is sort of changing either so the ultimate goal is here to how these monomers find each other for interaction, and the idea is the temperature will change the diffusion so that these molecules bump on each other in order to get into these lower stages, right? That's the idea. Temperature would play a role there so that these molecules meet each other.
So that's a good point. So then maybe I will talk about this just in the next slide. So here you are talking about also the kinetics of formation of this versus the equilibrium properties. Okay, so that's something which is maybe not trivial for you, is that when we describe that, we do not care about the kinetics of formation of this. We assume that we are at equilibrium, so meaning that these things are stable or not. But we don't care too much so far about what is the way they will be formed. So, but we know that in the real life, they have to be formed. Okay, so, and that's, I will talk about this later. And so temperature will, of course, have an impact on the diffusion of this molecule, so on the kinetics of formation of that, but have also an impact on the entropy. So the higher the temperature, the more entropy, the more entropic the system is, and the particle wants to spread around. It's not necessarily related to their speed or to their diffusion. It's just that they prefer to be spread, okay? And that's what I described here about the temperature. In the next slide, I will talk about kinetics, which is an interesting part, but it's not necessarily related. You do need to describe the kinetics here to talk about the stability. I mean, that's something which is important in physics. And so to continue, yes, you will.
In a simpler way, the entropy is proportional to the KBT.
Yes.
So actually, KBT is cancelled out in this equation. In a simple case, the entropy is proportional to KBT.
Yes.
Okay, so which means that in this equation in chi, KBT is actually cancelled out.
No, because, I mean, this is, in this equation, basically chi is a ratio, okay, between the entropy by thermal fluctuation,
okay,
KT, and the energy. Of course, the energy, because maybe it's a free enthalpy that you are talking about. It's again, it's very subtleties in an actual system. If you want to quantitatively describe that from an actual system, E, again, it's integrating a lot of internal degree of freedom because you are looking at interaction between two molecules, but it may depend on the relative orientation of that. And you have also an entropic and a contribution to this. And usually here you have this E is an enthalpic part, which does not depend on temperature, minus an entropic part, which depend, and that will cancel out. Okay, but okay, this is interesting, but that's let's say step 3 or 5 in the maybe in the description of the system. Yes.
There is a phi log phi term which is separate than chi term.
Yes.
We will decide the entropy. That is a chi. So this is just the enthalpic part.
Yes. Yes.
So I was just thinking of this effective temperature that. So I was just thinking of this effective temperature that he was saying. So in the context of these chromatin condensates, these are mostly like, for example, in the super enhancer, they're like active regions, right, in some sense. So should I think that the fluctuations there are more, and therefore, if I were to define an effective temperature, it would be higher, which would lead to a lower chi here, or should I not think in those terms?
This is a good question, and I don't have any clear answer on that. Yeah, the intuition would say that if you have more activity, your system have a high kind of effective temperature, which is higher. So, but maybe the interactions are stronger. So it's yeah.
Okay.
So again, it's a balance between the two, and so, but I think experimentally we have absolutely no ideas about this and how to define really clearly an effective temperature versus a thermal fluctuations. What we know, for example, for the diffusion of chromatin, and maybe you can comment, is that when you remove ATP, the coefficient of diffusion of chromatin decreased a lot. So meaning that what the diffusion we see is somehow ATP dependent strongly, okay? How? I think it's a good question, and I think very few people are trying to address that. So maybe if someone wants it, it's a very nice question, but it's a difficult question, I think. Yeah, no, exactly. Yeah, it's a good point. Yeah, you have different time scales in the system, and likely that's the diffusion we're observing at the second or minute scale does not reflect the exactly the thermal fluctuation, but at very small time scale, you're right. I mean, what you should observe is more thermal fluctuation, but experimentally, we are more capturing the dynamics at, let's say, second scale, which likely depends more on the ATP-like properties. But that's a good remark.
There's another thing. Since you're discussing equilibrium thermodynamics, here phase separation will be completely like one big phase, one big area of one type. Whereas in biology, you have finite size droplets. So where does that come in? Will you be discussing that later, where the finite size, not this complete phase separation?
Yeah, you're right, you're right. So maybe that's something, yeah, that's something that I didn't say first, is that when I'm talking about phase separation, it basically that your red molecules will be divided into two phases, a rich phase and a light phase, okay? There will still be some red molecules outside the condensate, okay, that will diffuse. But a lot of them will be in the condensate. But again, in the condensate, there will also be some blue molecules. Okay? So it's not like you have a pure phase of red versus a pure phase of blue. You still have some blue in the red phase and red in the blue. And with the proportion of blue in the red phase or of red in the blue phase depends again on all these microscopic details. In this what you're right is that in the Flory-Huggins theory, this is very sharp. So you have the green phase will be very green, let's say, and the blue phase will be very blue. Okay. But in more advanced theory, depending on the details, you can tune that actually. Okay, so just to finish that, is that so if chi is not strong enough, you will never see a phase separation, whatever the proportion of blue and red. Okay. And at some point, when you reach this kind of critical point, you may see phase separation depending on the concentration. So if I am here, for example, if the concentration, this is the concentration in red, is too small, basically the energy is not strong enough and entropy is somehow winning, and so there will be no phase separation. At some point, as you increase the concentration, this is this line, which is called the binodal. Okay. The condensate starts to be stable. Okay, so the systems prefer to be in rich phase. And then you have a domain here between these 2 points for a given chi, where condensation is stable. And at some point, when you cross a line here, as you increase the concentration, the system does not phase separate anymore. Okay? And so there is a line, which is called this binodal, where the condensate is stable by itself, and it's a first order transition to answer the question here. It's like, it's the same type of transition somehow, if you want, the same class as the freezing of, when you freeze water somehow, you have this kind of first order transition. At some point, you have a mixture of liquid water and solid water. Okay. Water is a very complex phase separation, so you cannot describe this by that, but it's the same principle. You have both entities at the same time. It's the same here. Another properties of this kind of very simple phase separation is that this saturation concentration. So in this region where the condensate is stable, okay, so when you have this formation of this condensate, the concentration of the red molecules in the light phase, so in the phase of blue, remain constant. It does not depend on the global concentration. So basically, if I am here in my system and I adding more and more red in the system, I will basically just increase the size of my condensate, but the concentration of my red monomers in the blue phase will remain constant. Okay? So that's another, if you remember, potential function of phase separation is to buffer the concentration of one entities. If you need a molecule to have a very fixed concentration at low level, okay, for some region, it's good actually to make droplets of this molecule with a high concentration somewhere, because you will control the concentration in the rest of the system, whatever the fluctuation of the global number of proteins in the system. So that's a potential, another function of this condensate, is actually to control the concentration in the place where there is very few molecules. All right, so this is, for example, a simulation of this model in 3D. Yes?
Can you go back to this buffering thing?
Yes.
So let's say if I have a particular protein of a very high concentration, so you're saying a cell can have these both things simultaneously going on. Some condensates which will have low concentration of protein, simultaneously other place high concentration as a sequestration of molecules to keep them as a stored protein.
So you will not, I mean, again, in this simple theory, you will not have it will not be condensate with low concentration. You can have this, but then you will have to think about multi-component phase separation. And but in this simple theory, the concentration of these molecules outside the condensate, outside, okay, there will still be some molecule diffusing in the system outside. This concentration is controlled. It will be constant, whatever the total number of molecules in the system. Because if now I'm adding new molecules in the system, they will go in the condensate in the rich phase of this molecule. They will not populate more the region where this molecule is very rare. Mm-hmm. So it could be a way, if somehow, if the function of this molecule requires a very low level, but very constant and very well controlled, that's potentially, again, I'm not sure there is even a biological context where this has been proven, okay? But again, we like to make hypotheses. Potentially, that's a function for this type of condensate. So you agglomerate a lot of proteins there, so potentially to make also functions to increase the concentration locally, but for free, you somehow have a control of the concentration of these molecules in the rest of the system, which will be constant and buffered to all the fluctuation. It might have a biological function, I don't know, but.
Okay.
So in the graph, I'm kind of becoming hard for me to understand, like when you increase the concentration of a particular solute, I understand that it might grow, the condensate might grow from low to high through the axis in one X line. But after that, you are saying that the condensates will not form. It's in this particular explanation, I would think that it should grow enormously until we have the whatever concentrations we have. But what makes the condensate to dissolve at higher concentration?
So it's not really a dissolution. It's quite subtle, I have to say. Okay, you have to think about the symmetry of the system here. Okay, when you have a condensate, okay, in this region, for example, which is clearly defined, let's say, I will have a phase which is highly rich in red and a phase which is highly rich in blue. Okay. And that's the phase separation. You have really these two phases, one rich in blue, one rich in red. Here, for example, where the global concentration of red is very high, right? I will only have one phase rich mostly in red. But the blue molecules that are traveling inside this ocean of red molecules will not form a condensate by itself. They will be spread, okay? So of course, if the concentration is very high, well, in the extreme case, I only have red molecules in my system. Of course, I can have a kind of condensate, okay? But if I have a bit of blue molecules now in my system, very slightly, this blue molecule will not phase separate from the red. They will move freely inside the red. Okay, so that's why we say that the condensates are more stable. You have to think symmetrically, okay, somehow. And same here. Here you have very little red, but a lot of blue, okay? And again, in that region, the red itself will not form a condensate. Here it's just the symmetric behavior, okay? You have a lot of red and a little of blue, but the blue itself will not form a condensate, okay? It will be spread in the red phase because, again, you have a lot of red, okay? So that's the small subtlety in that, where the concentration here. So I don't know if I continue or if I stop. 5 more minutes to talk about kinetics, because I think it's important. Okay, I'm described here. This is the binodal is basically, so this blue region is a region where we have phase separation, blue and red. But it's completely independent of kinetics on the way these things are formed. How the experiments are done usually. You mixed two entities together and then you look at what's going on, okay? So this is a simulation of the models that I show you in 3D, where I first assume that I have a mixture of red. This will be red and blue. Well, actually, this is only blue here. And only red, sorry, and the blue, I'm not showing the particle in blue. Okay, I'm just showing the particle in red, sorry, and actually the red, which are blue, sorry. So my red molecules here are blue here, okay? The blue particles here are white here. And when you do see a red thing here, is the local concentration, okay? I'm just coloring the beads as a function of their local concentration. If this is blue, it's just that I have just one isolated monomer around a lot of other molecules. If this is very red, it means that this is surrounding by a lot of the same type molecules. And it's a random mixture, and then at t = 0, I switch on the interactions between molecules. And then you will see the kinetics. So we are really modeling here the diffusion of molecules and how it goes. So it happens very fast. And what you can see, I'm going again. So here there is nothing. And rapidly you have nucleation of droplets that grows, that fuse eventually, that decrease. Here, this one just disappears. And this one, you have the fusion of these two big molecules. So there is a dynamics happening here. And at the end, I will have one gigantic phase, highly rich in red. I was talking about this model make a rich, very rich phase. But you still have concentration of molecules in the light phase. So that's what is showing it to you. And actually, what you can show is that in this gigantic region, there is two different families of kinetics, of how this, at the end, you will anyway, at the end with this big phase. But there is two pathways, two ways of forming that. This way here, it's, let's say, the classic way that we observe all in our kitchen or when likely to happen in the biological framework. Okay? And this is mostly in this metastable region here.
So in your model,
yes.
Why everything is accumulating in this particular droplet, even though they started with sometimes at similar concentration?
Yeah, everything the concentration is more or less homogeneous at the beginning.
So what I mean is, in biological system, in the nucleus, the nucleation may happen at many, many places.
Yes.
Eventually, from this model, you are saying that many of these droplets will fuse, coalesce, and form this one big droplet. Spatially, in nucleus, where would this happen would be important.
Yeah, this is a good question. So what you see here is actually the succession of mostly three types of process. One is nucleation. So you first form many droplets at different times. In that case, completely randomly, because it's just if you have local fluctuations. So if somewhere in my system I have, by chance, a higher number of molecules there, I will nucleate a droplet here. But this may happen at different places in my system, and so I will nucleate many things. That's why you see many droplets that nucleate at the same time. Then you have a second process, which is how these big droplets meet and coalesce together, okay? And there is two ways of doing that. This is typically shown here. So the first step is exactly this one. It's called diffusion limited growth. It's based nucleation. So basically, you just initiate the formation of this condensate locally, and this is just driven by diffusion. Only the molecules that arrive in the condensate will grow the condensate. But at some point, you can see here, at some point, you form several condensates. And so how do they coalesce at the end to form these gigantic things? This is the next step. So this is the first step of the fluctuations. And this is the two other way of growing the condensate. Because again, at the end, the equilibrium properties should be one big phase. And you have two ways. One, which is a more intuitive way. Okay, the one that, we think is what is called Brownian coalescence, that these two molecules will diffuse in the space and at some point will meet, and then they will form a bigger droplet. Okay? And that's what you observe at the very end of the movie, actually. Playing the movie again. Forget about, but at the end you have these two droplets that diffuse slowly to the other, and then they meet, and then they will absorb and make a bigger droplet. Okay? But something that you may know is that if an object is big, its diffusion becomes slower. Okay? So at some point, let's say in the cell, you have this very big phase. It will take non-physiological time for them to meet because the diffusion will be so slow, and for crowding, for many things, they will be basically stuck in their region. Okay? Another way of this to happen is by something called Oswald ripening. It's a kind of strange behavior that's not intuitive at all. Okay? Is that the small droplet will evaporate in the bigger one. So gradually, they will lose molecules, and via diffusion through the media, will populate the bigger one. And that's also something that you see in the movie. If you remember, you have a droplet. This one, look at this one. It does not diffuse. It just shrink, disappear. This is the process. It's because it's more stable for the molecule to go there, and that's the kinetic pathway will be that small molecule will evaporate. It's like evaporation, actually, in water. You evaporate, and so then naturally, thermodynamically speaking, it will be better for them to populate than the...
So is it because of the reduction of the local density of the molecules in the solution?
So the balance between the two depends on the tension properties of the droplet, mostly. Okay, it's a good point.
Danny, I have a question here. So I didn't understand this thing. So in case of this ripening process and the coalescence, so this rate which is proportional to the cube root of t, the rate, it seems like with time, it's not rate, maybe radius, right? That is the radius of condensates.
No, this is the radius of the condensate as a function of time, in fact.
So in case of phase coarsening, so there in Oswald ripening that you said, so there it's like diffusing away from a bigger condensate and then it's merging to a more bigger and making it bigger and bigger. So why this rate relation or the radius relation with respect to time is similar in between coalescence and this? So isn't it, if it's a smaller molecule, the diffusion rate would be more, so that it can be higher chance to get into a fuse to a bigger condensate compared to if two bigger condensates has to come together and meet?
So the scaling law is the same. So maybe just for the others, okay? This is a way to represent the typical radius of a droplet as a function of time, monitoring from this type of simulation. And you can look at how this radius changes as a function of time. And for example, at the very beginning, it scales as a square root of time. Okay. Physicists, you may know since the beginning of the week, like scaling laws and how this because it tells you something about the intrinsic nature of the physics. At some point, it changed the behavior. It becomes slower, okay? And the slope became 1/3, okay? But by chance, I don't know, but the scaling law is the same. What changed between the two is a prefactor, and I will not have the time to discuss that, but the prefactors are different. So basically, and the prefactor, the details of the prefactor depends on the details of the model you are using. In the Flory-Huggins theory, Oswald ripening is somehow favored compared to Brownian coalescence. There is still Brownian coalescence, as you have seen in the movie, but most of the coalescence is driven by Oswald ripening because the surface tension is quite high in this system.
So maybe because of lack of time, we can discuss over coffee. So Daniel, you could close in 1 minute if you want to say something more, and then we'll break for a coffee.
And just another movie, which is nice, is, and just for the last minute while I'm talking, is the other pathway for kinetics, which is called spinodal decomposition, which is when you are deep in the stability, thing is very unstable, and so you will not have exactly the same pathway. And this is a simulation in 3D. It's not, we didn't do it, but to simulate the way you form, which you see it's very different, okay? Again, so just to conclude, this is a very simple theory, and if I had 1 hour more, I will show you that in the nucleus, actually, this theory is too simple, okay? Because in the nucleus, you don't have only single entities, just one phase separation between water and oil, or between your proteins and water, okay? You have multi-component phase separation. So you have different entities that are interacting with each other, and this changed a little bit the picture, okay? The message here is that do not, even if this is the first time you show this, a lot of biologists, sorry for that, think that phase separation happened this way, okay? Like these very basic properties. And if they do not observe these basic properties, they would say that it is not phase separation, that's what they're observing. Maybe it's true, okay? Maybe this is not phase separation. Maybe this is something else. But again, in vivo, the complexity makes that you have multi-component phase separation. And so the intuition that we may have here, which maybe for you it's the first time you saw this, but for physicists it came from a long time, and for biologists from this cooking intuition about oil and water, we all have this intuition about phase separation. But if you don't see these hallmarks of phase separation, it doesn't mean that what you are observing is not phase separation, okay? Because again, you have different components interacting with each other, it changes the picture. And I will end up with that.
Okay, thank you, Daniel. Please thank Daniel.
And happy to discuss anything of that later.
So Daniel is around for next few days, so please catch him and please discuss with him.